Just survived a leg day. Now, let's do part 2. So, in the other part we covered a bit of starch, half of polysaccharides. Now, let's talk about water potential, because this is kind of important. So, what is water potential? Let's get into it. So essentially, water potential is just kind of a fancy way of scientists saying, oh, the concentration of water. Why? I don't know, they just like to be fancy. So here I'm showing you a diagram of water. Pure water, there is nothing in it. It is completely pure water, which is literally just distilled water. But this is water, pure water. There is nothing in it, no other minerals, nothing. It is pure water. And the water potential of this is obviously going to be the highest it can possibly get, which is 0. That's the value we give. It's the highest that the water potential could possibly get. It is as pure as possible, and it is shown by this sign, this Greek sign. It kind of looks a little bit like Aquaman's trident. It's kind of cool. It's called PSI. It's a Greek letter, and it shows you in kilopascals, in pressure, what the water potential is. So let's say I grab my pure water, and I do a bit of a silliness, a goofiness, and I add some salt. Just an example, it's a solute, I dissolve salt in it. What happens? So I dissolve salt in it, what happens? It's no longer pure water, okay? I have now contaminated it, I have now put salt in it, it is now impure. So, as you'd expect, the water potential was at its highest because it was pure water, but now it's gone down. The water potential has gone down because it is now contaminated, it is now less pure. And we'll give it a random value, let's say negative 100, okay? By the way, the water potential cannot be above 0, okay? So it can't be positive, it always has to be either 0 or negative. And it rarely is ever 0. I mean, when you see water out in the wild or something, being a bit of a goofer, it's rarely ever pure water, okay? It's kind of rare to come by. There's always usually something dissolved in it. So usually we don't really expect it to be 0 most of the time. Alright bro, so let's talk about our favourite friend, osmosis. Right, we've got a silly definition incoming, so remember this bro. It's a net movement of water particles from a high water potential to a low water potential through a partially permeable membrane. But again, we'll get to that soon, but just understand that osmosis is a process, it's a passive process that moves water molecules from a high water potential, so closer to 0, to a low water potential, so more negative. So let's grab our 2 solutions here, okay? And let's say we do a silliness and mix them together. What happens? So, of course, we have our pure water on the left and our salty water on the right. Now, of course, the water is not going to like being in that pure water, in that high water potential, because again, remember, osmosis is where they move from a high water potential to a low water potential. So they're gonna be like, nah, fuck this, and they move over to the lower water potential, they move over to the more negative part, and they do this until they reach something called equilibrium. Now, what is equilibrium? So eventually, once osmosis has occurred and the water molecules have traveled from one point to another, higher water potential to low water potential, eventually the water potential kind of both become equal to each other. So you have an even spread of water molecules on both sides. And due to this, there is no more overall movement. There's no overall movement of water particles. And here we say it's reached equilibrium, so it's isotonic. Now, the reason I'm bringing up this topic of water potential is because it's actually related to carbohydrates. So let's say I grab my pure water again, and I add some monosaccharides and disaccharides, and as we know, they are soluble. What happens? It lowers the water potential. Now, let's say I grab, my longer polysaccharides, my starch, my amylose, my amylopectin. I dissolve it in water, my pure water. What happens? They are insoluble. They don't dissolve in water, so they don't change the water potential. They don't affect the water potential. Bro, learn that. They will come at you with this in the exam, bro. AQA is ruthless. But then again, I kind of say it like it's one guy. I don't think it's one guy. I think it's like a team of people. I don't know, man. I don't know. What would it even stand for? Andy, Quagmire, Andrew? I don't know, man. I don't know. Alright, bro. I'm showing you how this applies to a real scenario, real context. So, we have here a cell in the blood. And I'm pretty sure this fella just scoffed down on some donuts and now he has some glucose molecules floating around in his bloodstream. So, that means his bloodstream is quite low in water potential, right? He has a high glycemic index, which means the blood sugar, and his blood is quite, there's a lot of sugar in his blood. So what happens? Low water potential in his blood, and his cells, compared to the blood, don't have as much of a low water potential, so they have a higher water potential. So what happens, bro? Why don't you tell me? Yeah, go on, I'm waiting, bro. Come on, tell me. Alright, bro. So what happens is, due to osmosis, which is a movement of water particles from a high water potential to a low water potential through a membrane, well, what happens? It moves that way, and water comes out of the cell. It comes out of the cell, and what happens is the cell starts to shrivel up. Because it's lost so much water, and now the water potential has kind of changed, it's lost so much water, and now the cell has kind of just shriveled up. Now I'm showing you a guy, and his bloodstream has the same water potential as his cell. So they're both negative 97 kilopascals. What do you think happens, bro? Tell me. If you said equilibrium, you're stupid, mate. Nah, I'm joking. You got it right. It is equilibrium. There's no net movement of water particles, so it's equilibrium. It stays like that. Now, what would happen if I somehow magically took those glucose molecules and I stored them all into the cell? What would happen, bro? Tell me. So, as you can see, we're doing that, and the blood is getting more pure of water because it has less dissolved solutes, which is the glucose. So I'm doing that, the water potential rises in the blood, the water potential goes down in the cell because it's less pure. So I'm doing that, bro, and what happens? You now have a difference in the concentration gradient, you have a difference in water potential. Now what happens, bro? It's just osmosis. It moves from a high water potential to a low water potential. And because water moves into the cell starts to swell up and get bigger, kind of like your mum. And then it just bursts. Alright, bro, here I'm showing you another dude, okay? He has amino acids, he has disaccharides and monosaccharides in his blood. But his cells also have the same water potential as the blood, okay? So, nothing complicated, nothing really has changed here. And what if I was to do a silliness, right? A goofiness and add some polysaccharides into his cell for the fun of it, okay? Now, I did say starch is a plant polysaccharide. It's used by plants, so I can't really add starch here, but let's just say for the sake of it, I add a polysaccharide, okay? I add it into his cells. What happens, bro? Tell me. So remember, bro, they're at equilibrium. They have the same water potential. There's no overall movement of water. What happens, bro? I put the polysaccharides into a cell. They don't dissolve. They are insoluble. And guess what, bro? They're still at equilibrium. Nothing moves in, nothing moves out. The cell doesn't burst of water. The cell doesn't, shrivel up. Everyone lives happily ever after. So what if I was to grab the polysaccharides and throw it into the blood? What would happen, bro? Well, the same thing. It doesn't dissolve in the blood, and equilibrium is still reached. The water potential is not affected. No osmosis happens, and again, we live happily ever after. So I'm pretty sure you can kind of figure out why. Why does an organism use polysaccharides as storage and not monosaccharides? Because you might think, oh, monosaccharides are easier to use, right? Because they're already broken down. Well, that's the reason they don't use it, bro, is because of osmosis, because of water potential. So your body, right, the organism has figured out like a life hack, like it's figured out a life hack. It's like, yeah, bro, what if I transform it into polysaccharide, right? They're proper big brain, they're smart. So they use polysaccharides as a storage molecule instead. Too smart, bro. Alright, this bloody bollocks has gone on too long, so I'm gonna cover the last bit of polysaccharides in the third part. I'll see you there, bro.